हम्म, नमस्कार।
नमस्कार नमस्कार।
नमस्कार नमस्कार।
आप ठीक है?
बिल्कुल
Namaste
ठीक।
from me too.
नमस्कार नमस्कार।
How are you? How are you?
I'm good.
We finally caught you, right? Thank you. Thank you so much. We're looking forward to your talk.
Yeah, I'll just
Tamil Nadu. So they wanted to find a scapegoat. So leather industry deals with the goat, they found a scapegoat. So they decided to make me the director. It so happened in 95 they closed 35 factories. And within 1 month I was made acting director. The Supreme Court waited for how long? On the day of my appointment as the full-term director, Supreme Court closed 400 factories in Tamil Nadu. So a good omen for the industry. Somebody had become the director on the same day industry closure. We were on a plane. The industry fellows from Supreme Court all beaten up, and I was coming from CSIR. We were in an Air India flight. We took a meeting in the plane. Well, we'll solve the problem. So we need to find a technical solution to this problem. There's a legal closure because of pollution problem. But we have to solve the problem of pollution for them to open. We needed to find a SNT solution for it. So we've been telling industry people all the time, don't do this, don't do that, you'll pollute, you'll pollute. So we talk about don't ecology. I moved to the common the denominator, do ecology, not don't. To protect the ecology, do this. So tell them positively what you need to do. So we talked about do ecology solution. So what I did was, I said a lot of things which are not correct. First of all, how does the water authority come? Now, government of India, government of Tamil Nadu has to close down. I cannot go back and say, look, give me 9 months, I'll solve the problem. 9 months, Supreme Court won't wait. And the Tamil Nadu government has to impact. So I went and told the Chief Minister, sir, we'll have technology solution, we'll solve the problem. And he found that maybe I have some convincing system. Then he sent a Solicitor General of Tamil Nadu how to approach the court. We said if you close down these factories, 2.5 lakh people lose jobs. That means 1 million people will lose livelihood. So they'll be dependent. You'll create a law and order problem. In order that we avert the law and order problem, we'll give CLRI and the NEDI lab 9 months to solve this problem. They gave not 12 months, 9 months. 9 months to solve this problem. So Supreme Court gave 9 months. And we also suggested Supreme Court appointed a committee, a judge, to monitor this implementation of 9 months. Within 9 months, they will deliver. So if the progress is not taking place, they can close down anytime during the 9 months. So we had all those systems. 148 colleagues of mine went and worked for 9 months.
Hello, everyone. Good morning, India and Indonesia, and good afternoon, Japan and South Korea, Hanyang. Very welcome. Good morning, yes. So very welcome to Cafe Series 100, and this is the moment I want to say thank you to you all to let us reach this time. It's just not that we organized, it is because of you guys. You attended and kept coming, and that's why we were motivated to go on this Cafe Series. And today we are celebrating Cafe Series 100 in AIST and in our satellite cafes. And up on my screens, I have thank you for all sister dialabs, IIT Delhi to begin with, Dr. Sundar and his team. As you know, our homepage is maintained by him and his team all the way through, and all cafe reports as thanks card are uploaded there every time we have cafe. IIT Guwahati, large number of students been attending. Triple ITD, Jaspreet has moved from IIT to graduated and moved to Triple ITD. Her team joins every time. GNDU, we see the big team today, and you have been with us all the times. Thank you so much. Sikkim University, welcome and thank you once again, and Manipal University. So today, the theme of Cafe, as we call it as Grand Cafe, we have distinguished speakers. And just in case, if you guys do not know about saffron, I'll be doubtful if you don't know, but you may not know that these beautiful, threads come from Crocus sativus flower stigma. And you can imagine how difficult it is to collect all that numbers that we see in our food and elsewhere. And I'm sure everybody from India knows about the puja thali, right? That's incomplete without saffron. The tikka we put on during puja is saffron and rice. So it's a sacred and auspicious spice used in Hindu rituals and puja thali. And for people who do not know about puja thali, they probably know about saffron from food. Biryani, we enjoy it all the time in parties, right? Those orange sprinkled rice are saffron rice. Then we have saffron sweet rice during festivals, and none other than ras malai that we enjoy every time in parties as a very favorable dessert is saffron. If you don't even know about that food, you definitely know about saffron cosmetics. It's very, very becoming very, very popular. Girls, I think so many of you, the young girls, must know about saffron as a toner, as a revitalizer, and freshening, reagent in the modern cosmetics. And now it's a time to know saffron as a supplement because it has so many bioactivities which are shown in my screen. I wouldn't read them in the interest of time, but it is becoming as a very popular supplement, and not only the supplement, but also drug. And especially for the old age related pathologies up on the screen, Alzheimer's and Parkinson's and several others. So I think with these, my introductory slides, you're all geared up to hear more depth saffron, right from the cultivation to the bioactivities. And I, with these words, get into the program. It's also up on my screen here. So we'll have our director, our director, actually, I would first say, because Dr. Motomichi Doi has been our director until March 31st, and he actually has been with us all the way through for DAILAB. And from April 1st, he has become our Deputy Director General. Congratulations to him, and I will invite him to, for the opening address.
Thank you, Lenny-san.
Yes, Doi-san, please.
Can you hear me?
Yes, we can hear you. I need a mouse.
And hello, everyone, and Namaste, India. So, first of all,
can you give me a mouse?
Professor Manol Nesari from AIST and also Professor Manol Dar from the ACSIR and Dr. Arun Sharma from NECTAR, and all the invited speakers and participants. So as Enu said, I'm Motomichi Doi, and I'm currently serve as the Deputy Director General of the Life Science and Biotechnology Department in AIST Japan. So until the end of this March, so I held the role of Director of the Cellular and Biotechnology Research Institute, where AIST-INDIA DAILAB is positioned within our department. Indeed, I have been associated with the activity since its beginning in October 2013. So when we designed the agenda for the activities, we emphasize the training components as important as the research itself. And some of you may be familiar with our workshop held in our AIST Tsukuba campus, such as the imaging workshop and other workshops. So today, our cafe, has been our regular activities connecting around 6 to 7 sites in India, and for around 6 weeks. I feel proud today that we have reached series 100. And I congratulate the entire DAILAB teams, especially Renu-san and Sunil-san, for their consistent efforts over the years. In this Cafe 100 celebration, we are honored to welcome again the Professor Manol Nesari, Senior Advisor of Minister of AIST, Professor Manol Dar, Director of ACSIR, and Dr. Arun Sharma, Director General of NECTAR. So, our AIST has joined research agreement with the AIA and ACSIR and NECTAR, and we are now preparing to undertake extensive research and training projects soon. So today's cafe topic, SAFFRON, is quite important for both research and commercializations. Saffron is well known for its delicate aroma. We Japanese also enjoy saffron rices and also other saffron sweets. However, I have not realized it's one of the most expensive spices in the world. Indeed, we should know the global attention should be paid to meet the cultivation challenges and expand its therapeutic scope in nutraceutical and pharmaceuticals. I welcome you all with today's cafe and look forward to having good and active discussions. Thank you very much.
Thank you very much, Dr. Doi. Thank you so much for being with us and letting us reach the day today. I thank you from the bottom of my heart. Next, on the schedule, we have Ambassador of India to Japan as a scheduled special address. However, we just got a message that he has an urgent meeting and he might show up later. With this, I'd like to move on and request our next special address, and this is by Dr. Manoj Nesari. Dr. Nesari, it's so good to see you here. Few lines about Dr. Nesari's introduction. Well, he's trained as an Ayurvedic practitioner, and I was so pleasantly surprised to see that he has a doctorate on policy research on international laws related to traditional systems of medicine. Not only that, he topped the merit list of UPSC commission exams at his time. That's an additional star in his crown. And his current core role, I would say, he does many things. His core role is the advisor to the Ministry of Ayush to the Government of India as the key person in integrating Ayurveda as a main course treatment in public healthcare. And he's head of the International Cooperation Wing of Ministry of Ayush, and we hope that we contribute to his roles by collaborating projects. Dr. Nesari, I welcome you and I request you for the special address.
Thank you, Dr. Renu, for this kind introduction. Thank you very much. And I'm also thankful to Dr. Renu as well as Dr. Sunil for inviting me for this cafe. Good afternoon to all of you, those who have joined from the Japan. And yeah, now even for India also it is now good afternoon. So good afternoon, Dr. Doi. As all of you know that the saffron, it is used widely in all the traditional systems of medicine. It is used in Ayurveda also. It is used in Unani as well. It is used in the Persian medicine, in many systems of medicine. The saffron is widely used. And in Ayurveda, there are references for the saffron from thousands of years. And saffron was being cultivated in India since ancient time. And that's why what actually Dr. Renu mentioned about the puja thali, that's why the saffron is there. The saffron is also considered auspicious. It has a very pleasant aroma as well and a very beautiful color. And beyond that, it has many medicinal properties. So from the traditional medicine perspective, all the substances in the world, so they are divided into mainly two components. One is the hot, another is the cold. So hot means those generate the heat or they are good to control the various the issues which are caused by the cold property, because there are two energies, the cold and the hot. So the saffron actually helps in generating the heat in the body. And this is the beauty of the nature, that the saffron is cultivated in the cold climatic conditions, and that is very useful to generate the heat in our body and to maintain our body temperature, and at the same time to to cure the various diseases which are caused by the kapha and the vata. So it is good for that. And if we talk about the specific conditions, so saffron is very good externally as well as internally. For external use, as you know, it is widely used in the cosmetics, especially in the blemishes and the scars and other deep pigments. So it is helpful in clearing the skin and the complexion of the skin. So that is widely used, this saffron. At the same time, the saffron is also used in various kind of headaches. So the saffron is also used in the form of paste, and it is applied over the head. At the same time, the medication, the oil which is made up from the saffron, so that is instilled in the nostril, and that is useful in curing the headache, whether it is migraine, whether there are different kind of headache. So it is used in the various kind of headaches. It is also used in the mental disorders like the depression and the other of the antipsychotic. So it is also used in that, and for that purpose, again, it is used in the form of NECTAR. NECTAR is the nasal administration, so it is formed in that formation. The saffron, it is used for the gynec conditions also, especially when there is the difficulty during the menstruation. So that time the saffron is used, and saffron improves the menstruation, so it is used in that. Saffron is also used in the conditions like the difficulty in micturition. Suppose if there is any obstruction in the urination, so it is also used in the dysuria conditions. And for that purpose, the saffron, it is used along with the honey. So saffron and honey, so this is actually mixed together, and it is kept overnight, and then it is to be consumed in the morning. So these are the references which are available in the ancient classical texts like the Sushruta Samhita, and we practice routinely, and that helps in clearing the urine, and that helps especially the patients like the prostate and other things. It helps them in getting getting the symptomatic relief. More research is actually required on the various aspects, that whether actually it acts on the alpha receptors or which kind of other receptors, how actually it helps. So that kind of research is required. So at the same time, it is also used in the cardiac conditions. The saffron is used. There are various preparations of the saffron in the ancient classical texts, whether it is the kumkumadi ghrutam and kumkumadi tel, the nasal administration I mentioned. The saffron can be used as a medicine as well as the food, and especially in those countries where the Ayurveda is not recognized as a traditional system of medicine. So in those countries, say for example in Japan, we can have the research and the evidence on the saffron. We can use the saffron and saffronless formulation as a nutritional supplement or a health supplement, and we can help the people to get rid of their various elements. So there is a lot of scope for the research as well. And I'm sure that the collaboration between the Ministry of Ayush and the AIST Japan, so we can take up various projects. So we have developed a concept called as the Ayurtech, that is the integrating Ayurveda and technology. And for this, we have collaborated with the various IITs. So with these IITs and with the help of the volunteers of Ayurveda and the ITI Jamnagar and the AIST, so we can have the research on any of the selected topic of the mutual interest, and we can we can develop some good projects, and that will be very useful for the community. So there is a lot of scope for having this collaboration, and I am really thankful that you have got so many experts from the various organizations, whether it is CSIR, whether it is from the other institutes, and this is a good gathering that we can have the good thought process. It is a Samudra Manthan. So with this Samudra Manthan, we can achieve something very good, like a Navneet, that is the butter in the form of the research and the various projects. So thank you very much.
Thank you. Thank you. So much information. You told us what saffron is used for. That's really, really wide and very valuable information. Thank you so much. And thank you for being with us. We know how busy you are and how much you're running around. So still you managed to be with us. Thank you so much.
Thanks.
Okay, so it's time to invite our first invited speaker, Dr. Professor Manoj Kumar Dhar, from basically from Jammu, and now he's in Delhi. So a few lines about his introduction. I wouldn't be able to say too much in the interest of time. Professor Manoj Dhar is geneticist by training, and he served as Vice Chancellor of Jammu University and currently the director of AcSIR. I'm sure everybody knows AcSIR, the largest academic institution for PhD level research in India. Some 6,500 students are enrolled with AcSIR, and we have leader of those right in front of us and going to speak to us for next 20 minutes. Right. So besides this heading the huge, large organization, Dr. Dhar has trained large number of PhD students, and he's fellow of National Academy of Sciences, National Academy of Agriculture Sciences as well, and he holds many prestigious awards and honors. With these words, I welcome you, Dr. Dhar, and please share your slides, and next 20 minutes is all yours.
Thank you, Professor Renu. First of all, let me say that it's a great pleasure to join on the 100th cafe, DAILAB-CAFE, and today is a very important day and a historic day, not only for AIST and India DAILAB-CAFE, because it is the 100th session, but it's also a historic day because today is the 13th foundation day of ACSIR also. So,
oh, wow.
Greetings to DAILAB as well as greetings to ACSIR and all the associated centers of ACSIR, that is all the CSIR laboratories, all the ICMR laboratories, and other DST and other ministry laboratories. So greetings to all of you, to all the stakeholders, all directors, students, everybody. So let me say that I am extremely pleased to be here this morning, and thank you so much for extending this invitation to me. And Dr. Doi, the Deputy Director General of AIST now, and Dr. Sunil Kaul, Dr. Renu Wadhwa, Dr. Manoj Nesri, Advisor to Ministry of Ayush, and I am told that His Excellency Mr. C.B. George, Ambassador of India to Japan, is also likely to join. So welcome to him as well. And I also extend a very warm welcome to Professor Pratap Pati, whom I know is a close friend, and other speakers who have joined today, Dr. Aparna, Dr. Nashiman, Ashraf, Dr. Fayaz, Dr. Colin. So, and all the students, all the distinguished scientists who have joined this cafe. So on my behalf, on behalf of ACSIR, a warm welcome to you. So without now spending too much of time on the introduction, I'll straight away go to, I think, should be possible.
Yes, I already have you permitted. Just share. Yeah.
So are my slides visible? No.
No, I see myself.
Just a minute.
So it's coming up. Yeah.
Yeah. Slides.
Yes, we can see.
Yeah. Okay. So let me begin the talk, and I have titled it as Understanding the Biology of Saffron to Strategize Its Production in a Changing Environment. As already extensive introduction about saffron has been given by Professor Renu as well as by Dr. Manoj Nesari, and he talked about several utilities of saffron. So I will actually talk more about the biology of saffron, and of course I cannot stop myself in saying that it's a very important plant and a highly useful plant. So, but I will start my presentation with the saffron myth, legend, and history. Mythology says that Crocus was a handsome boy in love with nymph Smilex. Hera, out of jealousy, transformed Crocus into flower Crocus and Smilex into climbing plant Smilex. Smilex is a plant, Crocus is also a plant. But legend says that Greek god Zeus slept on a bed of saffron. Alexander the Great, on reaching Kashmir, settled his army on a plain. The following morning, he saw an ocean of violet flowers around his tents and under the hooves of his horses. The flowers could only have been saffron. History says that archaeological studies in Crete, Cyclad Islands, and mainland Greece have revealed the presence of saffron in all these areas. Crocus was known in Minoan period in Crete 3000 years ago. On the wall of Minos Palace at Knossos, Crete, 1700 to 1600 BC, are frescoes depicting crocus gatherers. The period when saffron was introduced into Kashmir is not precisely known. Evidence from the oldest historical treatise of Kashmir, Raj Tarangani, written by Kalhana in 12th century, indicates that saffron was present in Kashmir even before the reign of King Lalitaditya in 750 AD, and it is known as Kumkum and Kesar in Sanskrit and Kong in Kashmiri language. As Dr. Nesri already spoke about evidences from our ancient texts that saffron was used since ancient times in India. Now, it's a mystery. The sources are ancient artwork, timeline of saffron usage. If you see the images painted using pigments derived from crocus, they trace back to 50,000 years. Prehistoric cave paintings in northwestern Iran, today's Iraq, they have used crocin, which is derived from saffron, for painting. Then information about origins, images that depict saffron flowers. You can see on the right, gatherers of crocuses on wall paintings, which I was speaking. You can see they are gathering saffron. It is about 1700 BC. So first evidence that saffron was being used. Now, despite many occurrences across artworks of ancient world, the first written evidence of cultivated saffron is only found about 350 to 300 BC in Historia Plantarum by Theophrastus. Now, what is saffron? Crocus sativus is the name of the plant. It's a perennial herb. Dried stigmas of this plant, you can see those stigmas, 3 stigmas, they constitute saffron. It belongs to family Iridaceae. Genus Crocus has about 80 species and all are propagated through corms. It is known under cultivation for spice since last 500 years in Egypt and Middle East. It is the costliest spice in the world because of the labor required for its cultivation, harvesting, and handling. Each flower has 3 stigmas. One stigma weighs about 2 milligram, and 1,50,000 flowers or 150,000 flowers must be carefully picked one by one in order to produce 1 kg of spice. Its high value makes saffron the object of frequent adulteration, and fraudulence, and it is highly important in human health. So far as its genetics is concerned, it's a triploid sterile plant and it is propagated by corms. It doesn't produce any seeds. And it's vegetatively cultivated, as I said. That is the reason it maintains genetic uniformity. All these corms are genetically uniform. And because of that reason, since there is no variability, it impedes any genetic improvement. It has a long history of cultivation. Wild progenitor is a subject of speculation. It has 24 chromosomes. That is why 2n3x24 would mean it's a triploid. There are 5 diploid species which were considered possible ancestors, but recently Schmidt has established Crocus cartwrightianus, which is a diploid, as the parent of Crocus sativus. Genetically speaking, molecular biology speaking, it has a large genome size of about 3000 megabase pairs, which is a huge genome. So far as its cultivation is concerned, in Kashmir, it is primarily grown in Pampur region, but also now the cultivation has been extended to other areas, and it's also grown in Kishtwar region of Jammu. But now, as you will be known in maybe the last speaker today will speak more about how the cultivation has been now extended to northeast region and also to Himachal Pradesh. So just a glimpse of how the flowers look like when there is flowering somewhere in October, November. In terms of production, saffron is cultivated mainly in Iran, India, Spain. There are other countries as well. The world's total annual production is about 205 tons per year, and Iran produces about 80% of it, 160 tons. In Kashmir, it is generally between 8 to 10 tons, but it's mostly consumed within the country. And I am told, and I have the data to say that India is importing saffron. In terms of production trends, in Iran, 43,000 hectares of land is used for saffron cultivation. That is why they give 90% world production. In India, that is Kashmir, about 3,600 hectares is used. Although there has been a trend in terms of decreasing of the land area. As you can see, in 1997, it was about 5,000 hectares, but in 2015, it has come down to 3,600 hectares because of many reasons. Source is J&K Agriculture Department data. And the most important part in saffron is the stigma and all the properties which Dr. Nesri has also spoken about, and Professor Renu also told about, that is because of the apocarotenoids which are present in saffron. Now, the stigmas are the points of accumulation of apocarotenoids formed as a result of oxidative cleavage of zeaxanthin. That is, these products, the apocarotenoids are quercetin, crocin, picrocrocin, and saffronal. And being seed sterile, propagation is exclusively through vegetative means, that is through corms, and it has hysteranthus flowering. That's what we biologically call a situation when flowers appear first and then the leaves will appear later. The pattern of metabolic utilization of starch in crocus corms bears a general resemblance with the pattern of utilization of reserve polysaccharides, because after first year of flowering, next year the corm will start producing small daughter cormlets. They are called as daughter cormlets. So corm size and apical dome diameter exhibit a positive correlation with flowering in saffron. Now, this is the schematic representation of apocarotenoid biosynthetic pathway. It starts from geranyl diphosphate, then to phytoene, carotene, and then a stage comes when there is bifurcation. It results in the formation of trans-lycopene or cis-lycopene. And this cis-lycopene, this channel, it goes for alpha-carotene and lutein, and this later channel or this another channel goes to beta carotene, from beta carotene to zeaxanthin, then to finally these crocin and quercetin and picrocrocin and saffronal. So one of the efforts which many people have been doing, and in our lab we have also been doing this work, that trying to see that how this can be channelized towards one side only, so that we have more quercetin production or more crocin production. So in terms of chemistry, carotenoids, as I told you, apocarotenoids, these are crocin, which gives color to saffron, then saffronal, which gives aroma, and picrocrocin, which gives bitter flavor. And this is how we started traditionally. Jammu is not a region where saffron is grown. Of course, in Kishtwar region it is grown, but not in Jammu because of the climate. It's a subtropical climate, but we could actually get to saffron and grow it and started cultivating it in the lab conditions, and we were successful in getting flowering and everything, as is being claimed by many people that they are able to get flowers and produce saffron. So it's true, it can be grown if you have the combs. The first year you will always get the flowering. Now we actually started our study by classifying these combs into different stages of development. You can see these 4 stages. First to 4, they are underground, they happen in the soil, and then later 5th to 8th stage it starts coming out, and 8th is the last stage where the flowering takes place and flower gets opens out. So we isolated, similarly, we classified combs into 3 groups based on the weight. For example, we took 2 to 4.9 gram, then 5 to 7.9, and greater than 8 gram, because has been shown that generally the comb size has to be at least 8 grams to get the flowering. So we thought, what all is happening in terms of molecular biology? Let's do that. Let's analyze that. And we picked up these genes, cloned these genes, sequenced them, and tried to understand their expression at various stages, as I showed you, and in different comb sizes. And we could see that definitely at the last stage, that is the 8th stage, there is very high activity of these enzymes and these products. And because of the expression of these genes is very high in these tissues which are up at the 8th stage. Similarly, we could find that the ones which are more than 8 gram, there this activity was definitely much higher. So thereby confirming, yes, it is true that all these activities are taking place. The expression of these genes is very high in the combs which are more than 8 grams in size. Then we also tried to see the validation of the promoters, because ultimately if you have to manipulate or work with these genes and to get higher expression of these, then you have to take other studies as well. So we studied the promoters and tried to see that how we can enhance the expression of those genes by treating them with different kinds of excuse me. So then in terms of on the promoter activity, we also studied the effect of different phytohormones. We also did transcript profiling of transcription factors expressed as various stages of stigma development, and identified 22 transcription factor families which could actually affect the stigma development. These are the changes of expression pattern in some transcription factors at various stages of stigma development. Due to paucity of time, I may not be able to kind of give in details about all this, but you can always refer to the published data which is there. Then the conclusions are that expression profiles of some of the carotenoid, apocarotenoid pathway genes during different time intervals, we developed those relative gene expression studies during different time intervals of apical bud development revealed maximum expression of apocarotenoid biosynthetic genes during 8th stage of bud development, and there was low or negligible expression during other stages of bud development. There was sharp increase in expression of all genes from 6th stage to 8th stage during bud development, suggesting their possible role in apocarotenoids. In terms of flowering, many people have studied and identified flowering genes in saffron. I'll not focus on those. We specifically wanted to see what is happening at the microRNA level. So we took flowering and a non-flowering plant in order to understand which microRNAs or which transcription factors are actually involved in terms of flowering. So we have been able to identify, this is last year we published this in Protoplasma. So you can refer to this paper and see that we have done an extensive analysis to identify the microRNAs in the flowering and non-flowering tissues, and these are the novel ones which we have identified. and the ones which are already had been reported earlier. But I don't think there was any extensive study on microRNAs of saffron. So that's what makes this study very important. And we have been able to identify some specific microRNAs which have showed higher expression in non-flowering tissue, thereby suggesting, and we proposed a model to show that how these microRNAs are actually regulating various flowering genes, and that is perhaps helping the plant to either produce a flower or not to produce a flower. Because we could see that there are some combs, they, even if the size is right, everything is correct, but they still are not flowering. So what could be the reason? That we have tried to speculate from here and hypothesize from here based on the data on microRNAs. Now, in terms of genetic manipulation, we thought that since saffron is a triploid, so it is not producing any seed. So genetically, what could be the best way of getting the seeds? The best way of getting the seed would be to double its chromosome number to make it a hexaploid. And we made efforts in that direction. We were successful also to produce the combs which are double the size of the combs which are there normally. And we could show with other studies like stomatal analysis, chromosomal analysis, that these were indeed hexaploids. But unfortunately, these plants, the combs could not actually grow beyond this condition. There was sprouting, we could get sprouting from colchicloid daughter comblets, but they were not able to grow further. These experiments were conducted in Kashmir University Botanic Garden. So finally, we were left with no choice but to abandon this, because ultimately we could not actually get any, I would say, sprouting beyond that. So technically, yes, hexaploid is possible, but if it would have been successful, perhaps it may give us some seeds, and that would revolutionize actually production of saffron. But yet, not every experiment is successful. So sometimes you have to see the failures as well. But we haven't left this. We are continuing with this process. So maybe in future we may get some success in this area. Then we were also able to establish a protocol for disease-free production of disease-free combs. We could in fact increase the size of the combs, and finally we could transfer them to the field as well. So it's a complete end-to-end technology which was developed for production of disease-free combs. As you know, in combs there is always a sometimes there is comb rot, and that can cause a lot of devastation to the combs. So this is a technology for production of disease-free combs. We also developed a system for callus production, somatic embryogenesis, and gene editing using CRISPR-Cas. We published it around 4 years back. The efficiency is very low, but then we could demonstrate that yes, CRISPR-Cas or gene editing is possible. And now we are going ahead with this. And since maybe you are aware that saffron is a very slow growing plant in cultures as well. So we are facing a lot of difficulties in this, but we are going ahead with the gene editing process. Now, in terms of genome, as I already mentioned, it's a huge genome, 3000 mega base pairs, and we have initiated a project 2 years back to get the whole genome sequence assembly. And we sequenced the saffron genome using Illumina platform. A total of 1.2 GB draft genome sequence has become available and has been assembled now. Now, complete whole genome resequencing employing nanopore platform has also been achieved. We are trying to integrate now this genomic, proteomic, and metabolomics data for identification of regulatory pathways. And I'm sure that this is going to change the scenario. Of course, there are reports of genome sequencing from other countries as well, from China and others. But then since it's a huge genome, everybody is finding it very difficult to finally get the genome of that size, sequence of that size. So I'm hopeful that in future we'll be able to do much better in this case. So I think that's all, keeping in view the time shortage. But then I would definitely like to thank Department of Biotechnology, DBT, Government of India, for generously supporting our research in saffron. 3 projects end to end, they have been supported by DBT, and I am really grateful to DBT. Then students, 2 students got PhD from Saffron work, Archana Bhatt and Sudachip, and 2 postdoctoral fellows. They conducted research in Saffron, Dr. Sonal and Dr. Munish. And we have a lot of people who have actually contributed to this work. It's not one person's job. It has to be done in a collaborative manner. And we have Professor Nikhil from Nehu, Professor Ejaz from Srinagar, Ashwini from JNU, Mukesh Jain from NIPJR JNU, and Dr. Tanushri Kaul, ICGB, and Professor Sanna Kaul, who are in the team working towards the betterment of saffron. So thank you so much for the opportunity. I hope that I didn't take much time. Thank you.
Thank you so much. Thank you for the extensive knowledge that you have delivered today. And one burning question we can have right now, and I request others, if you have questions, please write it on the chat, and we can address to those at the end. And if anybody wants to ask right now, just raise hand. I wondered, when we are enjoying biryani and rasmalai, we never thought of that there is so much biotechnology going into this. And the younger students here need to think about the cosmetics, right? So there is no question right now, so we'll keep the questions at the end. And with this, I once again thank you, Dr. Dhar, and we move to the next speaker. So we already realized that so much biotechnology is required in this. And here I invite Dr. Aparna Pati from IHBT. A few lines about Dr. Pati. I know Pratap Pati. So when I say Pati, I imagine him. I have to say this is Aparna Pati. Okay. She is Chief Scientist and Head Biotechnology at Institute of Himalayan Bioresource Technology, IHBT it's called. It's CSIR institution. And her research interest is biofertilizer for sustainable agriculture and waste management. And she's mentoring large number of students, has completed several projects very successfully, and transferred technologies to industries, and have several awards to her credit. Okay, Dr. Aparna Pati, please share your slides, and 15 minutes is all yours. Tell us about saffron more. Yes.
Thank you, Dr. Renu. And congratulations to complete, DAILAB team for successfully conducting this CAFE series. And this one is 100 series. And greetings, my greetings to all. And you have chosen a wonderful topic for this particular, program. So saffron, I think it is one of the best topic that you have selected. So my respect to all the seniors, and I express my gratitude to the entire team. And though I am working in the area of biofertilizer and sustainable, agriculture. So I have worked a part of my work in saffron, but that is not my, whole soul research. And speaking after Dr. Manoj Dhar is very difficult, who is, actually an encyclopedia on saffron. So I'll just try to, explain as to what little I have done in saffron. And it's a wonderful topic. Thank you. Thank you for giving me this opportunity. So as you know, the Dr. Manoj Dhar and the esteemed speakers, Dr. Nesri, he has already mentioned about the economic importance of saffron and how it is important for our particular society, as it has wide scale application in cosmetics, then pharmaceutical industry, in food, in color. So this is actually called as a red gold, and true to its economic value. So if you see the global market, it is around 600 million, and it is constantly increasing, say at the pace of 7%. So we see that there is, larger scope of economic advancement with respect to saffron day by day. And it is important to increase its cultivation, because we have seen that the cultivation is in decline, particularly in India. So the domestic market, that is the Indian market, if you see the need is of 100 metric tons annually, while the production is around 7 metric tons. So our country is heavily dependent on imports, So it is the, I think, responsibility of the scientists who are working in the area of saffron as to see how this particular, cultivation can be increased. So that is the major challenge. And we have seen decline in production. This is due to disease. Disease is one of the main cause, because you see the cultivation area of saffron is confined to the Jammu and Kashmir region, particularly the Pampore region. And due to and years of monoculture, there has been lot of disease incidents. The disease buildup is very high in this soil. So whatever the daughter corms are being produced, they are also not as healthy, so there is constant decline in production of healthy corms. So that we have to keep in mind. Further, to add to the peril, there are rodents. Then, of course, due to anthropogenic activities, there is shrinking of land, which Dr. Dhar has already mentioned. So as a result of which, it is also a labor-intensive farming, and getting labor is one of the most, challenging, you can say, issue in agriculture. Further, there is climate change. We cannot predict the climate that it will remain the same, or there will be sudden fluctuations of high and low temperature like that. So that also, decreases the production considerably. So we have to anyway face this challenge and march ahead. So the major, step that, India needs to take is extend the saffron cultivation. And we have seen that some, efforts are already being made in this particular area. Now saffron cultivation has started in Uttarakhand, Himachal Pradesh, and also to Northeast. We'll be looking into it in little detail. Okay. So but thing is that if we say that, okay, we have to increase the saffron cultivation, so which would be the area where we can actually pinpoint to make an initiation? So particularly our institute is working in the area of modeling, though my group is not directly involved with it, but we are getting the expertise. So we have a group who, focuses on this aspect, and they have done the MaxNet modeling. And by, compiling the data of temperature, precipitation, radiation, wind speed, vapor pressure, and other things, they have actually mapped out which are the areas which could be suitable for saffron cultivation. And depending on that, they have mapped as to the high area, like probability area, medium, low, like that. So taking a cue from that particular, map, we have started cultivating in certain regions of, Chamba, then Mandi, then Upper Himachal, that is Lahaul and Spiti. Then I'm just, because I'm working in the area of plant growth promoting, rhizobacteria. So I'll just confine my talk to that. So we have seen that this particular agriculture, field, which is adjacent to the Kangra, our, institute, repeatedly this particular field showed good production of saffron years after years. After 3 years of study, because we know that, microorganisms also play an important role in shaping the yield. So we have isolated bacteria, that is the from this particular soil, and around 60 bacteria were isolated. So they were pure cultured, and systematically all the bacteria were checked for their plant growth promoting attributes. That is, the common traits are phosphate solubilization, siderophore production, IA synthesis, potassium solubilization, nitrogen fixation, and deaminase activity. So we have screened all the 60 bacteria with respect to these traits, and then narrowed down to some of the best, that were around some 14 bacteria. Then what happened is, after their in vitro assessment, we wanted to check how this bacteria will behave under different temperature regime, because we always say that, okay, the field, the lab results with respect to PGPR application is good, but when we take it to field, the most of the bacteria actually fail, and we say that biofertilizer not working, good enough. This actually happens because there should be stringent, check assessments of bacteria has to be done in lab. So what we did, we checked the potential of these bacteria at different temperatures as to how they will behave. So we, classified them into low temperature, mild low temperature, then normal temperature, and high and very high temperature. So then again, we screened all these bacteria, 14 bacteria, with respect to their in vitro, that is the phosphate solubilization, nitrogen fixation, attributes. And only those bacteria which could actually qualify this trait, we took it further. So this was not enough. After the in vitro assessment, we also went for pot study, because we don't know how a particular, plant and the bacteria will interact. So after this stringent pot study, we selected one particular bacteria, that is, FS2, and we found that it is performing extremely well with respect to its plant growth promoting traits under in vitro condition as well as under pot study under different temperature regime. Then, of course, we have to identify this bacteria. It should not be a pathogenic form. Then we went into its, like 16S RNA studies. We found it is bacteria Bacillus altitudinalis. The Bacillus altitudinalis is a safe bacteria, and that can be, further developed into bioformulations. Then we worked out the whole genome sequencing. Then I will not, touch because this is not particularly related to saffron. We said which are the phosphate, producing genes, then citrophore producing genes, and how they behave under different temperature regimes. So all, real-time studies was done with respect to genetic constitution and its behavior with respect to the particular bacteria. Then we also, checked whether this bacteria will be, tolerant to stressful conditions or not. We saw that it can, grow well from 4 to 50 degree. Then it can, sustain the pH range from 5 to 11, salinity up to 6% and 8% under different temperature regimes. So we were convinced that this particular bacteria is a stress tolerant bacteria. And if we apply them in field, the lighter, certain temperature fluctuations or the temperature from, cold to the high will not affect its functionality. So once we were convinced of its functionality, then we made entire, bioformulation. So from prestige, we went to flask culture, from flask culture to bioreactor. And now we have developed a commercial formulation of this, and we have applied to different crops. And saffron is one of the, crop which we have, studied and examined as to how it will behave with respect to its comb production, because comb production is very critical. In case you want to extend the cultivation of saffron, then you have to go for new and high quality comb. Then this is the major limitation, because even the combs which are produced in Kashmir are not very healthy, and there are not enough combs to take up, go for additional, cultivation in new areas. Areas are available, but planting material is not available. Okay, so we treated this particular saffron comb, which is, as Dr. Dhar has said, that only the combs which are above certain gram, that is about 8 grams, only they will flower. So we treated about 8 to 10 grams of combs with this formulation, and this study was conducted at Kangra region only. And we saw that there was good harvest. Interesting thing is that you see that in control plant, say 63% of combs were below 3 grams. Okay, because once you have this saffron harvest, then you will have different grades of combs. Some will be very small, some will be of medium size, some will be large size. So you have to do the grading of saffron. So with that, we graded the saffron into less than 3 grams, 3 to 5 grams, then 5 to 8 grams, 8 to 10 grams, and above 10 grams. An interesting feature is that the bioformulated applied, the combs which we, planted, out of that, 45 were, only 45% were less than 3, and rest were above 3 grams. And you can here see that, about more than 10 grams combs are around 12%. And in this case, control, it is only 5%. So 3 to 5, again, you can see and, feel that you get major, greater combs when you apply this biofertilizer. That means these saffron combs are actually positively interacting with the biofertilizer that we have applied, and the daughter combs which are produced have greater weight. So definitely when we go for cultivation, and because this is consumed either in food or for medical purposes, so it is advisable not to use the chemical fertilizer. It is better that we go for sustainable agriculture practices that are eco-friendly. So all these PGPRs are eco-friendly. So we just wanted to eliminate the practice of applying high doses of chemical fertilizer, and you can actually see that this particular PGPR bioformulation is affecting the positive development and growth of saffron combs. So after that, we also, included mycorrhiza, that is arbuscular mycorrhiza, and this study is underway. Here also you can see the positive interaction of saffron corms and PGPR and mycorrhiza. This third treatment, which has both PGPR and arbuscular mycorrhizal fungi, the corm blades and the weight of corms are more. So we are expecting to have a good harvest of saffron corm production when we apply, biofertilizer, whether it is, fungus, that is arbuscular mycorrhiza fungi, or PGPR, or a combination of both. So if we have to go for sustainable agriculture, then alternative to this could be a good alternative for chemical fertilizer. So this was all about the PGPR and the biofertilizer, which can positively contribute to the saffron yield as well as promote growth of healthy development of saffron corms. Now we also tried our hands in tissue culture protocol because that was a requirement of our DBT study. So here we went for actually to see cost effective protocol, because you can, if we wanted to transfer this technology to industry, the cost of corm production should be less. So here we have actually not almost you can say eliminated the use of all the growth regulator, or applied it at very minimum quantities. So this particular, we are still working on it. That is why I'm not reflecting the exact amount of concentrations that we have used, because this study is still underway. So here again, what we have done is we have shortened the protocol tissue culture cycle. So generally the commercial cycle or the literature available, it is more than 200 days, but in our case, we have reduced it to around 145 to 160 days. Okay, and it's a simple once you can say in each particular induction media, you just have to keep for 22 days. Then again in proliferation media, and in multiple shoot, you can keep it at number of times. Like you can continue it for several times, so that you don't have to go back to the saffron corms from the field. You can right away go to this corm formation media, and this particular industry can maintain their culture at multiple corm production cycle. And then you can see the harvested corms. So some of the corms weigh around 5.6 or you can say 5 grams, but this is not the general case. It is not that all the corms produced are of 5 grams or 6 grams, but they are generally average size is around 3.5 to 4 grams. So we are still trying to increase the size of the corm without using the expensive growth regulator. That is our endeavor. So we have achieved to certain extent, and these are some of the limited, you can say the highlights, that the cost reduction is around 3.24. The days are from 145 to 160 days, and survival rate is also very high. And it can serve as an excellent source of new corm production. And but then again, we are at present stuck because the limitation is the size of corms which we are getting. We are trying to actually achieve 6 to 8 grams corm in a single tissue culture flask. That is our aim. So this was all about as to my work. And as our institute is extending saffron cultivation, we are doing it in Uttarakhand as well as several places of Himachal Pradesh. And this model has helped us a lot, and we are getting a good quality saffron production, and the quality is at par with that of Kashmir. And these are some of the work that is being done with respect to indoor cultivation. We are doing this particular work. This Sawcast Kashmir University is part of our collaborator in a DBT project, and they have done good work in indoor cultivation. And they are getting good because it is a labor intensive crop, and if you have a simultaneous flowering, then this particular limitation will be overcome to a great extent. And then it is a two-step process. Again, you have to plant it in field. Under high density planting, it is giving a very good result, and maybe in coming 2 years we'll have functional workable solution for the farmers of our country. And these are some of the biological studies that are also the part of the DBT project. So we are still working on it and very hopeful as we are getting very promising leads. So this was all about, and I'm thankful to CSIR as well as DBT for funding this particular work. And thanks to all the participants and the listeners. Thank you.
Thank you. Thank you so much, Aparna. This was excellent talk. We actually wish you all the best for handling all this biotechnology for, saffron cultivation and beyond. Please, everyone, keep on writing questions on the chat. I have written some, and I request the speakers to also keep looking at the chat for the questions. And we move on to the third talk for today, and invite Dr. Nashiman Ashraf, CSIR Institute of Integrative Medicine from Srinagar. Are you there?
Yes.
Yes, good. Please
Good
share your
afternoon.
slide. As you
Yeah,
share your
sure.
slide, I'd like to read few lines about you. She's Principal Scientist, Plant Biotechnology Division again, CSIR, Triple IM Srinagar, and her main focus is characterization of pathway genes and their engineering to enhance quality of saffron. And she's going to tell us about generation of genome edited smart saffron with enhanced quality and stress tolerance. We need that all the time, right? Quality and stress tolerance. All you go. Next 15 minutes, please confine to 15 minutes. We can see your slides already.
Okay. So, good afternoon, everyone. Greetings from Kashmir, the saffron growing region in India. So, I'll be talking about generation of genome edited smart saffron with enhanced quality and stress tolerance. I will not go about introduction of saffron because previous speakers have already talked about it. I'll straight away come to what I want all the listeners to, know about, that in spite of being such an important crop, we have seen a declining trend in saffron cultivation, production, and productivity since last 20 years. And the major reasons for this decline are lack of irrigation, lack of quality planting material, and comrod disease. Like previous speakers, including Professor Dhar, mentioned, that we cannot go for breeding programs for development of some improved cultivar of saffron or climate resilient saffron because it's sterile. It does not produce seeds. It's propagated vegetatively through corms. So we have to depend on biotechnological intervention like metabolic engineering. Professor Dhar already showed this apocarotenoid biosynthetic pathway. I'll again come here. So lycopene, oh sorry. So lycopene cyclization, please mute your mics. Lycopene cyclization is the major branch point in carotenoid apocarotenoid biosynthetic pathway. If this lycopene is acted upon by lycopene beta cyclase, beta carotene is formed. Beta carotene is in turn converted into zeaxanthine. These two carotenoids are the major substrates for enzymes known as carotenoid cleavage dioxygenases, and these enzymes cleave carotenoids into apocarotenoids. Alternatively, if lycopene is acted upon by lycopene epsilon cyclase, it forms delta carotene, which is again acted upon by lycopene beta cyclase and converted into alpha carotene. This is the alpha branch pathway of carotenoids, and this is the beta branch pathway. And it's important to know that it's beta branch carotenoids which act as substrates for carotenoid cleavage dioxygenase and get converted into apocarotenoids. Like here, zeaxanthine is converted into beta cyclocitral and crocetine aldehyde by carotenoid cleavage dioxygenase 2. This beta cyclocitral and crocetine aldehyde are subsequently converted into crocetine, crosine, picrocrosine, and safranal, and these compounds are actually responsible for imparting color, flavor, and aroma to saffron, making it world's most expensive spice. Now, if we have to go for metabolic engineering of crocus, we need to make sure that we divert metabolic flux towards beta branch. That can be achieved by three approaches. One of the approaches would be if we increase expression of lycopene beta cyclase so that more of lycopene is converted into beta carotene. Another approach would be if we shut down expression of lycopene epsilon cyclase so that lycopene is not converted into delta carotene, rather the metabolic flux is shifted again towards beta carotene. Third approach would be if we change this lycopene beta cyclase in such a way that it binds lycopene but not delta carotene, and in that case again the metabolic shift would be towards beta branch carotenoids. I will be explaining these approaches one by one. However, I will start with talking about lycopene epsilon cyclase, because what we hypothesized is lycopene epsilon cyclase would have a better effect on diverting metabolic flux towards beta branch than overexpressing lycopene beta cyclase, because lycopene beta cyclase is present in beta as well as alpha branch. So we started with cloning of lycopene epsilon cyclase. So this is the sequence alignment of lycopene beta epsilon cyclase from crocus with its homologs from other plant species, and we can see it has two cyclase motifs, cyclase motif 1 and cyclase motif 2, which are responsible for activity of this enzyme. After in silico results, we wanted to know if the clone we have, if the gene we have cloned codes for a functional protein or not. So what we did was we cloned this lycopene epsilon cyclase in PGX40-1 vector, which is a bacterial expression vector, and then transformed this gene construct in lycopene producing bacterial strain, which we had obtained from AdGene. So when we transform this lycopene epsilon cyclase into lycopene producing bacterial strain, this pink colored lycopene was converted into colorless delta carotene, showing that yes, the gene which we have cloned codes for a functional protein. We also cloned or transformed this gene into delta carotene producing bacterial strain. However, delta carotene was not converted into any other products, it remained as delta carotene. This showed that lycopene epsilon cyclase from crocus is a functional enzyme and is a monocyclase. That means it converts lycopene into delta carotene. However, delta carotene is not further acted upon by this enzyme. Now we wanted to know which of the amino acids are important for activity of this enzyme from cyclase motif 1 and 2. So we did suspect analysis. What suspect analysis does is that it mutates in silico, mutates each of the amino acids and then checks its effect of this mutation on activity of this enzyme. So this is the mutation of amino acids from cyclase motif 1 and this is from cyclase motif 2. And the amino acids which when mutated affect or decrease activity by more than 80% were considered as critical. So we had many critical amino acids in cyclase motif 1 and cyclase motif 2. Next, we wanted to know which of these amino acids from cyclase motif 1 and cyclase motif 2 are actually involved in interacting and binding the lycopene substrate. And we found that out of all these amino acids, these 4 amino acids at these positions were involved in lycopene binding and hence were important for activity of this enzyme. Now, this was all about in silico results. We wanted to check these in silico results in vitro. So what we did was we mutated these amino acids one by one, and the mutated isoforms of these lycopene epsilon cyclase were again cloned in PGX40-1 vector and transformed into lycopene producing bacterial strain. And then we checked activity of this enzyme, these mutant isoforms. And here we can see the glutamate residue at 318 position, if mutated, resulted in complete loss of activity, confirming that this amino acid, this glutamate residue at 318 position is critical for activity of this enzyme. For further, deletion of cyclase motif 1 and 2 also resulted in complete elimination of activity of this enzyme. So this was about the in vitro results. Now we wanted to check effect of this lycopene epsilon cyclase in planta. So what we did was we constructed RNAi construct of this lycopene epsilon cyclase. We cloned it into RNAi vector. We prepared the construct and then transformed these crocus stigmas with this RNAi construct of lycopene epsilon cyclase. And then we checked, we used these transformed stigmas for HPLC analysis to quantify various carotenoids and apocarotenoids. Here we can see this is vector control, wild type, this is vector control stigmas. These are lycopene epsilon cyclase overexpression lines, and these are lycopene epsilon cyclase RNAi lines. And here we can see carotenoids like beta carotene, zeaxanthin, and crocin were all enhanced in lycopene epsilon cyclase RNAi lines, while as alpha branch carotenoids like delta carotene and lutein were reduced. However, we were not bothered for these alpha branch carotenoids. Our focus was beta branch carotenoids like beta carotene, zeaxanthin, and apocarotenoid like crocin. Now, as I mentioned, we can also enhance apocarotenoid content by overexpressing lycopene beta cyclase. So we wanted to compare effect of lycopene beta cyclase overexpression and effect of lycopene epsilon cyclase RNAi lines on crocin content, because crocin is the major determinant of quality of saffron. Here we can see wild type vector control, lycopene epsilon cyclase overexpression line, lycopene epsilon cyclase RNAi line, and this is lycopene epsilon cyclase, beta cyclase overexpression line. So here also we can see increase in crocin in lycopene beta cyclase overexpression line. Crocin is increased. However, it increase is more in case of lycopene epsilon cyclase RNAi lines, confirming that lycopene epsilon cyclase RNAi is better, candidate for metabolic engineering as compared to lycopene beta cyclase overexpression. Now, this was all about quality or like a quantity of crocin. What about climate resilience? We know abscisic acid is an abiotic stress hormone, and interestingly, abscisic acid is also an apocarotenoid. It's also a product of a beta branch carotenoid. So we quantified abscisic acid in these transformed stigmas, and here we can see abscisic acid was also higher in lycopene epsilon cyclase RNAi lines, suggesting that lycopene epsilon cyclase RNAi lines might be better tolerant to stresses. In order to confirm that, what we did was we subjected these transformed stigmas to various stresses like dehydration, salt, and UV stress, and then checked MDA content and radical scavenging activity. Lycopene epsilon cyclase, the RNAi lines had less MDA content and more radical scavenging activity, again confirming that these lines were more tolerant to stress. Another approach was to, what I mentioned here was to change lycopene beta cyclase in such a way that it binds lycopene but not delta carotene. That's the third approach we used. I think I have less time. How much time left, Renu? So what we did was we mutated.
As short as you can.
Okay. We mutated alanine at 126 position in lycopene beta cyclase and checked its activity on delta carotene. We found that it was not able to convert delta carotene into alpha carotene. However, beta carotene was enhanced. That means it acts on, it binds lycopene, this lycopene beta cyclase mutant isoform, it binds lycopene, but it's not able to bind delta carotene and convert it into alpha carotene. We again transformed this mutant isoform of lycopene beta cyclase in crocus stigmas and then quantified crocin, and we found crocin was much higher. It was even higher than lycopene epsilon cyclase RNAi lines. Then, both the previous speakers talked about how difficult it is tissue culture of saffron and transformation of saffron. We were not able to generate stable transgenic line of saffron. However, we transformed this mutant isoform of lycopene beta cyclase in Nicotiana tabacum to see if this affects growth and development of plant in a positive way. This is the wild type. These are the transgenic lines, and we can see by appearance on the transgenic lines are much better performing. We also checked beta carotene and zeaxanthin content in these Nicotiana tabacum overexpression lines, and we found that this mutant isoform of lycopene beta cyclase, when transformed in Nicotiana tabacum, had better carotenoid profile and could be converted into apocarotenoids, of course. So we also checked chlorophyll and electron transport chain efficiency. All were good in transgenic lines as compared to wild type. And finally, we confirmed subjected these Nicotiana tabacum lines to stress like dehydration, salt, UV, and methyl viologen, and then checked MDA content and radical scavenging activity. The transgenic lines were all having less MDA content and more radical scavenging activity, confirming that Nicotiana tabacum plants, which were transformed with mutant isoform of lycopene beta cyclase, were all having better growth and development and better stress tolerance. This means that this particular gene can be used for genome editing of saffron. It's a very difficult job. Professor Dhar, their group is also trying it. We are also on it. And soon, I think, we'll come up with smart saffron, which has better quality and stress resilience. So these are publications from our group. This is my group, and mostly the work done is which I presented today was done by Javed Mir. Thank you.
Thank you so much.
I'm open to questions.
Beautiful work, and congratulations and best wishes for the smart saffron. We don't see you on the video. We don't see you on the video.
Actually, I have some issue with my camera. That's why.
No problem, no problem. So if anybody has questions, please keep on writing on the chat. Actually, there is one, maybe you can answer or the previous speakers can answer. Hashim from Hokkaido University, he's asking, as carotenoids contain a lot of double bond, which can potentially undergo isomerization by light. So is there any difference between saffron grown in dark and light conditions?
There is no difference between saffron grown in light or dark. However, these apocarotenoids come to a peak in dark. Maximum content of these apocarotenoids in saffron is during midnight.
So does that matter that you harvest during dark?
Yes, that is why it's recommended that we should harvest saffron flowers early in the morning.
I see. All right. Does that answer your question, Hashim?
Yeah, partly, yes.
Right. Okay. All right. So thank you very much, Nashmeen. And we move to the next speaker. Please unshare your slides. And it's my pleasure to invite the next speaker, my friend and collaborator, Dr. Fayaz Malik, who has been with us all the way through the cafe. He has been the cafe speaker a few times as well. So a few lines about Fayaz. He comes from CSIR, Indian Institute of Integrative Medicine in Srinagar, and his interests are mainly on cancer therapeutics, and he has been using the natural compounds to test their anti-cancer activity, and a part of that also comes from the natural compounds, also referring to saffron. Because he's from Kashmir, everybody from Kashmir loves saffron, I know that. And here you go, Fayaz. Next 15 minutes is all yours, and we already see your slides.
Thank you, Renu. Thank you for the nice introduction. So without wasting much time, I will straight away go to my presentation. Is it visible to everybody?
Yes, it is. It is.
So a lot of powerful plant sciences presentations. We will take a break to see what saffron has, its role for health benefits. So most of my presentation is actually not my data, is the institutional data, where most of my data is from my other colleagues, and some of the data for the depression will be from my lab. So I will not talk about this thing. This thing has been already spoken out. So I will straight away go to my slide. So what our institutional mission project from CSR was to actually do the chemical characterization of this saffron, and based on this chemical characterization, we have made a formulation where we keep batch to batch to avoid the batch to batch variation. We keep 5 biomarkers in that in that formulation, which we have taken further for its understanding its role in different brain elements. So these are the characteristics which has been done in our lab. And next, I will tell you that this is the formulation. So we can see the total crocin level in this formulation known as IM141 is 63.4%, and total cross-series in the plant is 28% weight by weight. So several parameters have been observed to make this whenever there is a new batch of this extract is made, so that it's kept in mind that the new extract has almost close, or there is no change in the biomarkers, which biomarkers I have showed you here from M1 to M5. So having said that, so we have taken two diseases of brain disorder by way of focusing on. One is Alzheimer's disease, and other is depression. So because Alzheimer's disease and depression are the most common and challenging mental disorders in our society, and coming to the Alzheimer's disease, all of you might be knowing its background, and what I will tell here is like it is a 7th leading cause of death globally, and as per WHO, because the age, the people who will be crossing 60 years of age by 2050 will be almost 60 will be double by 2050, and so will increase the number of sufferers of Alzheimer's disease. So it is an age-related disease, and it causes dementia in people. So the biomarkers for this disease are like, there's an accumulation of that amyloid beta in the neurons, which cause the neurotoxicity that leads to neuronal damage, and subsequently leads to the disability of the person, and called due to Alzheimer's. So these are different stages of the disease. So what we did is my colleague, Dr. Ajay, and his group has actually tried to see whether this saffron formulation in its individual constituents can actually help to get this amyloid beta clearance from these brain cells. So what he has done, the important thing is that which his lab has actually understood that this saffron treatment actually promotes autophagy in the cells, and that autophagy is a process which is very important for clearance of these amyloid beta plaques from the neurons. So that's the so you can see here. So there's a lot of data. I cannot show you all the data. I have taken a few chosen slides here. So saffron treatment actually abrogated the AMPK pathway, which induces the autophagy in the cells. You can see there's a when we treat the mice with the saffron like III15533, so there was a phosphorylation of AMPK, so there was AMPK activation, there was a reduction in mTOR, phospho-mTOR. So when there's the inhibition of phospho-mTOR enhance the AMPK activation, so it leads to the autophagy. You can see here there's an increase in LC32, and there's also the increase in the other autophagy markers. So having said that, what we did next is, so they took N9 microglia cells, and you see when we give amyloid beta to those cells, you can see here there's an accumulation of amyloid beta in the cells. So when we give the rapamycin is an mTOR inhibitor which induces autophagy. So there's a kind of clearance from the amyloid beta from the cells. So when we give amyloid beta along with that saffron, so its effect was as good as mTOR, or maybe better than that. So then we also give the bepalomycin with the autophagy inhibitor. So there was no amyloid beta clearance here. So showing that the induction of autophagy by saffron actually leads to the amyloid beta clearance from the cells. So this is a graphical representation of the same experiments. So next is we took the 5XFAD mice, which are like known for the Alzheimer's disease models. So when they were given the saffron, so you can see, so there was a amyloid beta has drastically come down with the treatment of prostine in these mice, and you can also see there's brain levels there were down in the treatment of prostine, down regulation of amount of amyloid beta in the brain, and you can also see here 6C10 is the antibody which actually binds to amyloid beta. So you can see with the treatment of this mice with the prostine led to the you can see the diminished expression of amyloid beta in those brain cells. So after the mice were given the treatment for certain period of time. So next, so this is has been already published in neurosciences, ACS Neurosciences. The publication is already out in the journal. So now, when this saffron treatment was leading to the reduced amount of amyloid beta, it was also leading to the reduced inflammation in the brain. You can see the IBA1 is a marker for the microglia, which shows which is activated. The microglia gets activated during the inflammation. So you can see the expression of IBA1 is gone drastically down with the treatment of crostin in the animal models, showing that with the reduction in the amyloid beta in the new brain, it also leads to reduced inflammation in these in the brain. So that can actually lead to the next step is we actually did also pharmacokinetic studies of this formulation and this compound is M553. You can see it quietly crosses the blood-brain barrier. It is also available for a long time there, and when we got this brain tissues out of the brain after treatment, you can see there was a significant induction of MPK pathway and there was a induction of autophagy. So this is a mechanism of action which we show so far is which I have told that the amyloid beta clearance from the mouse in the Alzheimer model was because of the induction of autophagy, and same has been published in Autophagy Journal in 2017. And there's also certain other publications in this regard there. So next thing was if it clears amyloid beta, it's reducing inflammation. Was there improvement in the dementia? Because dementia is one of the consequences in Alzheimer's. So yes, we have seen that dementia is a huge global health challenge according to WHO. So estimated 50.2 million individuals globally will be affected with the dementia. So here, when we give the treatment of this formulation to the mice, and we used STZ memory deficient Morris water maze model, you can see they compared to this STZ model, you can see there was a reduction in latency time period of mice to find its target. So it took a lot of time in mice which was not given the treatment compared to this STZ model. When we give him the 50 mg and 600 mg of this saffron formulation, the change in latency time was quite significant, and mouse was able to reach its target in time. Though the same experiment was also observed on the 5xFAD mouse model, where you can see there's the number of targets the mouse was crossing was significantly improved with the treatment of the crostin, and the time of the target quadrants where he stayed in the target quadrants was more compared to the vehicle, showing that the treatment was actually improving its memory as well as and learning of the mouse. So coming to the next thing is which is very important is we understood is like saffron delays the brain and body aging by increasing ATP level, by increasing the energy states of level. So we have shown that this by treating the mouse for 4 months with the saffron, the the oxidative phosphorylation level were increased in the mitochondria of these mice brain, and that was actually a very important marker for for aging. So the more the energy, then the more the oxophosphorylation is there, the lesser is the aging. So we found that the oxophosphorylation were quite high with the treatment of saffron compared to the control, and these mice were quite healthy, and where they have a good memory and they have a good other parameters which we have performed, where showing that it delays the age in the mice who were treated with the saffron compared to the control. Another important biological activity of the saffron is it targets NLRP3 inflammasome. That is very important for this, a major contribution in inflammation. So this is the mechanism. The paper is already out. So this is a mechanism how this saffron actually reduce the inflammasome formation and reduce the inflammation. So it is one of the powerful anti-inflammatory agent. So coming from this, then there's another disorder is a depression. So depression is according to both the depression and anxiety, where two major conditions contribute in increased suicidal rate in the world. Globally, there are 280 million people who are suffering from the depression. So in India, an enhanced survey has shown that 150 million Indians are suffering from mental issue, and 1% of the population is of high risk of suicide. So similarly, the current treatments of there are the treatments which are currently available for the treatment of depression are, either are the serotonin receptor inhibitors or amino acid inhibitors. So 50 to 60% patients with depression do not recover fully from the antidepressant medications which are there, or 1/3 of them are, getting remission of the treatment. And 30% patients are actually resistant to the current treatment options. So the idea was to find out alternative way of plants which are nutritionally important and offer the same source for the intervention of depression. And saffron, because and the pathogens of depression, I'm sorry, I'm going to say the pathogens of depression. So here are the 4 different axes which actually are contributing into the depression. Is HP axis, monoamine hypothesis, neuroinflammation, and BDNF neuroplasticity. So we focused on this pathway, and BDNF is also known as the brain-derived neurotropic factor, is a biomarker for the treatment of depression. So when under the stress, it causes inflammation, and the BDNF levels goes down, and neuroplasticity goes down, and anxiety and depression goes up. So it has been also seen in the patients who have the suicidal tendencies or who are under serious depression, when these patients were given antidepressants and their brain were mapped, so it showed that the BDNF level were increased after these patients have been given some antidepressants. So the point here is the current antidepressants are also working by increasing the BDNF levels in the brain. So how BDNF works? BDNF works through TRKB receptor. So it needs neuronal proliferation, neuronal plasticity, and the more the BDNF is there, it's a better mood. The lower the BDNF is there, there's a it is opposite to, it's like sad mood, or there's a neurotoxicity rather that. So these are so we took BDNF as a marker to see what happens when we give the mice with the cell lines with the treatment of this saffron. So you can see here we give the treatment, the corticosteroid treatment, and you see the BDNF level went down in these cells. So DHF is a current clinical investigator, I think is a preclinical drug. So it also increases it. We took it as a positive control. So it increases the BDNF level in the cells. But when we give I153 is the saffron treatment, so the BDNF level were quite increased in these cells, which were actually pre-treated with the corticosteroid. So similarly, when we did the NMDA, treatment to these cells, the BDNF level went down. So when we give them saffron treatment, the BDNF level were, gone up again. So similarly, we did it when we saw the level of corticosteroid. So the corticosteroid levels were actually brought down by the treatment of the saffron. You can see here. And both similarly, when we in the NMD treated cells, the corticosteroid level were brought down by the saffron. So showing that saffron, while elevating the BDNF levels in these cells, simultaneously brought down the corticosteroid level. So maybe that will lead to the less neurotoxicity in these cells. So coming to this point, as I said, BDNF TRKB. So we try to understand whether this the pro-survival pathway get increased by the treatment of saffron. Yes, we have seen that the TRKB levels and the AKT levels were increasing with the treatment of the different concentration of the saffron, showing that this actually needs a neuroproliferation and in these cells. And the another thing was actually to see the effect of the saffron in the different models of the stress. So we took 2 stress models. One was COT-mediated stress model with the corticosteroid treatment to the mice daily for 21 days. We also did the CUMS, which is another model where we did 2 stressors per day. So after 26 a day, we did a behavioral test. Then we start giving them the saffron after 27 days. And we again, after 2 months, we checked their behavioral tests and then also isolated their brain tissues. We divided them into the we took out the prefrontal cortex, we took the hippocampus and tried to see whether saffron treatment can actually correlate with our in vitro data. So different behavioral tests we have done, like tail suspension test, force swim test, and you can see with the treatment of saffron, there was there was a glucose uptake by these mice who were actually treated with the saffron compared to the mice who were only stressed. In both the models, we can see that. There was also water preference test or the sucrose test in the when the mice were in the comes model of depression. So you can see the saffron treatment actually made them prefer sucrose rather than water when they were this mice who were treated with the saffron. So we can also see when we took the mice brain, you can see here the corticosteroid level were brought down by in the prefrontal cortex, hippocampus, and serum when the mice who were treated with the saffron compared with the fluoxetine and other insults like corticosteroid. The fluoxetine is a current antidepressant drug, SSRI inhibitor. So saffron, you can see the saffron, the treatment in bringing down the corticosteroid was as good as fluoxetine. Similarly, the BDNF levels were increased in these mice, and you can see the compared to the cord, the BDNF levels were quite high when the mice were treated with the saffron here. So it overcome the effect of the corticosteroid. So it was as good as the fluoxetine, which is a current antidepressant. The similar results we found in the hippocampus and prefrontal cortex in serum in both the models of CUMS and COT stress-induced models. And then we took out we subjected these prefrontal cortex to immunoblotting and see, and there was an increase in BDNF levels. You can see here when the mice, the mouse who were treated with the saffron, and there was also increase in the phosphoric, pro survival pathways. And similarly, they were in the COT model, which is a corticosteroid model, we found the same trend. The saffron actually enhanced the pro-survival pathways, BDNF expression, and TRKB expression in those models compared to the corticosteroid treatment in fluoxetine. So also we found that the saffron treatment also leads to the in prefrontal cortex, it is increased expression of neurogenic proteins like GLUN2B was increased compared to GLUN1. So showing that there was a increase in neurogenesis in those mice, which is a marker of neurogenesis. So showing that saffron treatment not only increased BDNF but also enhanced the neurogenesis in the mice brain. The same trend we found in the CUMS model, which is another model of depression, showing the same trend which we show in the corticosteroid model of depression. So in the nutshell, I want to show you that the saffron treatment in vitro and in vivo showed neuroprotective potential. It increased the BDNF level in the mouse. It improves the behavioral despair. It mitigated corticosteroid hippocampal prefrontal damage. So the formulation which I was talking about is there's a lot of data has been generated on this formulation, which is like PK, PD, and other. Currently, this formulation has been licensed to one of the pharma companies called Formenza. So the MOU has been signed, and the same formulation has been sold to this for commercialization. So there's another part of this formulation is it is used for Alzheimer's, has been already patented and is currently going for IND. Its IND has been also approved. Currently going for preclinical trials. Thank you. So this is a team.
Thank you.
So most of the data was from Ajay Kumar and his team, CMBRI, who was the chemist behind this. Yeah, thank you.
Thank you so much. Thank you, Fayaz, and congratulations. Let me mute the others. So, okay. So congratulations for good work, and thank you for, letting us know all this extensive work. There are a few questions Fayaz or the earlier speakers may also like to answer. Question from Hashim is, any studies reach into clinical trials? I think you partly answered.
Yeah.
Would you like to say something shortly?
As I said, we are currently in the preclinical trials of this formulation for Alzheimer's.
Right.
Yeah. There's a lot of data on that. Yeah.
Question from Dr. Gowda, again from Hokkaido University, is about what solvent extracts of saffrons has been used for the studies showing good results like in vitro and vivo. What solvent have you used for your extraction from saffron?
Yeah, it was 50% aqueous alcoholic extract.
Right. I hope that answers your question, Dr. Gowda.
Yeah, got it.
Okay. So there is a question from Anu Thakur as well, asking that many people using aeroponics to grow saffron. So it will be valuable to hear your thoughts on this growing trend. Would you like to answer, Fayaz, or?
I think Nashima will be the appropriate person to answer
Yes,
this.
like in saffron, by end of July or August, all the floral organs are developed. At this stage, if we take out the bulbs and grow in, aeroponics or hydroponics, there will be flowering definitely. But then for corm development, corms again have to go into the soil.
Okay.
So that they'll produce flower next year. Corm development will occur
it has
only
to go
in
to
soil.
the ground. Yeah.
Yes.
Okay.
Yes.
That answers. There's another question. Away from the disease-directed approach, what is your viewpoint about preventive role of saffron for healthy aging? I think you did show some data on mouse aging, but would you like to say something, Fayaz? Are you there, Fayaz?
Yeah, I'm here only.
Yeah. So effect on the healthy aging.
Yeah, it's healthy. Yeah, that's what I'm saying. So everybody should be, in the morning when they wake up, they should put some, some saffron in their drink and go to the office because it actually delays aging.
All right.
Yeah.
All right. So now you want to also tell us, has this to be Kashmiri saffron or it can be Iranian saffron too?
You better go for Kashmiri saffron.
Why? Why? Tell me why.
Based on its constituents, carotenoids and all that, the Kashmiri saffron has been shown has a better.
So it has been done and it has been shown that the Kashmiri saffron has higher amount of the bioactive ingredients.
It has been done in our lab as well. Yeah, we have shown it.
All right. What is the difference? What is the ratio difference? Like 20-fold, 10 or 100?
Precisely, I cannot say that, but we have found that our local saffron has a better constituents and better concentration of these compounds compared to the Iranian saffron.
All right. Hashim says it should be same as long as the molecular components are same.
It is same, but the point is that,
but the content is different.
But the concentration.
Molecular components are same, but the concentration
of the components is different.
Yes, Hashim. The bioactives are present in much higher content in the Kashmiri.
The desirable bioactive constituents are in higher quantity compared to this.
Right. Okay.
Okay. Can I ask something?
Sure, Gurchandni. How can you not ask? Yes. Okay, please.
When it comes to neuro, I get all my attention to it. So, Dr. Fayaz, very, very interesting, very huge amount of work you have done addressing AD and depression. So, my only concern is that we know Ayurveda is India's heritage, and we have several medicinal plants, ashwagandha as queen of Ayurveda, and then Tynospora as heavenly elixir, and we have saffron and many more. So what about taking them to a direction for testing on humans in an appropriate way, like having a formulation which works? Because our main obstacle is the availability of a quality formulation. So since CSR labs are doing such good work with the natural products, so I, being a retired person, always think whether we can take these two further. And for your work for healthy aging, I know AD, it causes, it's detected when already great damage has been done. Similarly, depression starts from acute anxiety to chronic and then to depression. So already damage is done. So can we really think of some strategy where we now take it to the clinical trials and the human studies so that we know how much dose and what quality product can work for healthy aging? That's my concern.
Well asked. Yes, Fayaz, there you go.
Yeah, it's a really good question, ma'am. So the point is, when we start this project as a mission project, CSR was generous enough to fund us. So the important point was in market, lot of companies, lot of brands keep selling these products, but the important thing is the reproducibility of these products. So in this saffron, what we did, we have every time we take a new batch of saffron, we purchase new batch of saffron, we characterize with our selected markers. We have taken 6 to 7 markers. So we have made sure that the concentration of these markers remains plus minus 10 same in using every batch. So if it is not same, then we don't take that batch. So we go back and get another batch. So that is one thing that we standardize, like in US they called it botanicals. So in that term, so we characterize them with a particular formulation. And now what we have done a lot of preclinical studies, because there's a lot of data here. We have done all preclinical studies to position this botanical formulation for clinic. So right now we have completed all the formalities which have been which led us to the filing the IND. So new IND for this formulation. So we have so recently we have filed new IND, and it DCJ has approved for this clinical trials, phase 1 clinical trials of this formulation. And the same path can be followed for others as well. It's comprehensive because compared to the in CSIR lab, so what we have done is we have followed all the requirements of DCGI, which are actually needed to take it from lab to the clinic, and which may probably was not followed in the university setup. So if these formulations can be, if these products can be actually followed in the same way, so definitely if there is a potential, they can follow the same path.
Yeah, very important because we have Ministry of Ayush also associated with this. So I think it should be taken to further higher level.
Most of the part of the studies from Ajay, it's my part's only from depression, so it was a whole team of CSR Jammu which was involved in it.
Thank you.
Right.
Thank you so much. Thank you, Gurcharandy. Thank you, Fayaz, and all the best. Before I invite the next speaker, 2 short questions are there. How about the saffron flowers? We are talking about saffron stigmas, which is saffron. How about the flowers? Would they be good or equally good or little good, whatever? So, can anyone answer the question? Nashmeen,
may I?
Yeah? Yes, please.
Like most of the medicinal properties are due to crocin, picrocrocin, and safnal, which are synthesized only in stigma. So flower cannot
Good
replace
enough. Yes.
stigma.
Okay. Another continued question is, can that be synthesized rather than extracting from saffron or flower stigma? How about the chemically synthesis?
Yes, if you remember my slides, I showed you that one of the saffron genes, we transformed it into bacteria, producing lycopene. So we have bacterial strains which produce zeaxanthin.
Right.
If we transform CCD2, carotenoid cleavage dioxygenase 2, in zeaxanthin producing bacteria, we can synthesize crocin.
Okay, last question before the next speaker is from Kazumi Hirano. He's asking, does autophagy induced by saffron also contribute to the degradation of tau protein, the other disease-causing proteins? I think Fayaz is the best person to answer this.
Well, the autophagy is a process where we can actually, the cell throws its unwanted material out of the cell, and so the same thing is
do you
here.
see the degradation of tau?
Yeah, we didn't see the degradation of tau, but we saw, the amyloid beta quantity inside the cell getting down.
Okay.
So that's we have, but it's a good
question. How about the extracellular beta amyloid?
Yeah, it was actually, as I show you, it was getting increased and eventually was thrown out through the phagocytosis.
Thank you so much. Yeah. We need to move on to the next talk. Please unshare your slide. And it is my pleasure to invite the next speaker from Northeast, Dr. Colin Renthele. Are you there? Please unshare your slide, Fayaz.
Yeah, sure. 1 second.
Yes.
It's taking a bit.
So as Dr. Collins is coming, please share your slides, Dr. Collins. Good to see you. Dr. Collins is Scientist D and Head Training and Extensions Division. They do lots of things, all related to application and outreach, and that the Institute is about. At the end, we're going to have some concluding remarks from our Director of the Institute, NECTAR. So now I invite Dr. Collins Renthele, and he's going to tell us the status of saffron in northeastern part of India. As you already heard that it is moving from Kashmir to the different regions, and we hear about northeast from Dr. Collins. Dr. Collins, please, 15 minutes is yours.
Oh,
Please,
hello.
yes,
Yeah,
yes, we can hear
am
you.
I audible?
You are, and put your slides on
Okay,
the
okay.
presentation mode.
Yeah. Is it visible?
It is.
Okay. I would like to thank to Dr. Vadia, and I'm sincerely thankful to the AIST for the kind invitation to deliver a lecture on saffron cultivation. It was really engrossing after seeing all the presentations of the eminent speakers and the respected members in the platform. And there are some students from reputed organization. The presentation that I'm going to give is a bit field related. It will not be on research. So the without much further ado, I'll just start my presentation. So the title of the presentation is Saffron Cultivation in Northeast India, a New Frontier. We are telling it as a new frontier because it is the first time that we are doing this saffron cultivation in Northeast India. Our Director General, Dr. Sharma, is there. He will be giving more insight about how this was done in Northeast India because it was one of his idea that we have taken by NECTAR since 2021. So we all know about the importance of these crocus flowers, where it is the most expensive spice in the world. It is called as a red gold. It is rich in bioactive compounds and all this high crocin content and all that multiple medicinal and pharmacological properties. So I will not go to that brief introduction. I will go directly straight to the problem. We all know from the presentation before also that in India alone, the annual demand of saffron cultivation is huge. I'm told that the demand is about 100 metric ton, while the production initially I thought it was 12 to 15 metric ton, but I saw the presentation given by Dr. Ashraf that it is 5 to 6 metric ton. So it is even lesser than what I thought before. So the demand of saffron, it comes only from the Kashmir region. So it is high time now that India explore alternate source to fill the gap, and that is Northeast. So we have developed some aim and vision. That is, number one is to assess the suitability of saffron cultivation in Northeast India, to introduce as a high value crop, as to provide livelihood to the farming community of Northeast region, and to develop standard practices for saffron cultivation in India, because the climatic and geographical condition is different in Northeast. We cannot always copy the way that we are doing in Kashmir, and to promote organic cultivation of saffron.
हाँ जी बोलिए क्यों
So
नहीं
why
ऐसा कुछ नहीं है हाँ क्यों
Dr. Collins, please unmute yourself. Dr. Collin, please unmute yourself. Yes.
Okay.
Yes.
So I would like to show to the audience that we have taken some 7 to 8 parameters to compare the geographical condition of Kashmir and Northeast. We have seen that the climatic type in Kashmir, in terms of the winters and the summers, we find in Northeast India also similar type of climates. Then the temperature also, we from 5 degree to 15 degree. There are many places in Northeast India, and proximity to glacier. We know that the Pampur region is very close to the glacier. So we choose those places which are also have some proximity to the glacier, like in Arunachal Pradesh and Sikkim. Then altitude-wise, we know that in Pampur area, the saffron growing area are in between 1,000 to 1,800 meters. Almost entire hilly area of Northeast India are in this within this range. We are having only one problem here, only the soil type and the rainfall, where it is not where it is not that similar to Kashmir. So this is the challenges that we are having. But in spite of that, through supplement and through other protective measures, we try to solve this problem. Then regarding humidity and growing season, we find many places in Northeast which are almost identical to Kashmir. So having said that, can saffron be grown in Northeast India? So we have taken around 6 states. There are two places in Arunachal Pradesh. One is called Mechuka, where we find that all the parameters like the winter temperature, the altitude, the soil, and the other factors are very suitable. So we identify those places as high suitability. There are two places in Mechuka. One is Shergaon. Then in Meghalaya, we find 3 places: Upper Shillong, Mairang, and Thangsning. So both all the 3 places, they have the moderate suitability, whereas the last one, although it is a low suitability, we tried in that area also. I will show you the result. And likewise in Sikkim also, we have 2 or 3 places that has similar climatic factors and geographical conditions similar to Kashmir. Like in Manipur, also Nagaland, 2, 3 places. I'll just run this one. Then we give the final ranking as the best candidates. In terms of all the parameters that we have given, there are 5 best candidates we have taken, which has high suitability. Then we classify them as moderate suitability for 5 places and low suitability in 4 places, as shown in the presentation. So the step that we followed for saffron cultivation is the desktop study through GIS mapping. Then after that, we went to the place and studied and took the soil and we analyzed the soil condition. After identification, we procured the saffron corms from Kashmir. We brought the experienced farmers from Kashmir to train us here in various places in Northeast India. So we are very happy to those farmers. So they are still in touch with us. Then we saw those saffron corms in various places. Then after flowering, we harvested and we assess the corms survivability and multiplication of the corms. So these are the area that has been taken and the collaboration that we are having with various state government. We have around 12 government establishments and NGOs whom we have collaborated. And we have covered about 63 farmers in our first phase of the pilot cultivation. So the quantity of corms that we procured from Kashmir was 27 quintals. So we have distributed those corms across 4 states. You can see that the number of the quantity of corms that has been supplied to various states as per their suitability. So we have noted down all the standard parameters of the locations that we have taken in terms of the latitude, elevation, temperature, average rainfall, the soil type. So these are the soil samples that we have collected. Some of them are not that very suitable, but in spite of that, there are some parameters where some suitability are there, but we continue to do on that one. So the steps that are taken for the cultivation of saffron cultivation, step by step, that has been taught to us by this Kashmir farmers, like from site selection, what type of soil is it required, then the depth, then because here the rainfall is very high, so we raise the bed. So all this type we modified as per the suitability here. Then the drainage. Drainage is one of the very important factor here in Northeast India because too much of heavy rainfall. It is more than 2,000 millimeters. So we have to do we have to search for some places which are a bit slopey in nature so that the water is not retained to prevent fungal infection and rottenness of the combs. Then we sorted the combs according to the size and we cultivated them separately. Those which are more than 8 grams, we find that they have more potential to bear or to bear the flowers. Then those which are more or less than 6 gram, although they don't bear flowers, they play an important part in multiplication and propagation and expansion of the combs. Then we follow all the steps that has been taught to us in terms of spacing and planting, the irrigation. When we talk about irrigation in Northeast, we are depending on rain-fed. So we shifted the cultivation period to a little bit later than the post-monsoon season, say in October. This is the place in Machuca. So the harvesting. Harvesting is also very important. Initially, we don't know how to harvest. But after some 2-3 days of the practical experience that we have, we find that if we harvest manually 10 to 12 hours after it has bloomed, so we get the freshness of the flowers. So regarding the post-harvest management, also we try to follow every steps of the way because it was our first or second time that we have been doing this one. So the drying process is also very important while doing the sun drying. So most of the drying we are doing we are doing sun drying under the shade, and the packaging is also very important. Storage is also very important. So every step we followed, whenever we have some literatures that has been shared to us by the Kashmir expert. So that these are the details of cultivation that we have done. So the quantity of combs sown during 2023-24 across 4 states of Meghalaya, Sikkim, Arunachal Pradesh, and Mizoram was almost 27 quintals. Here we can see the very interesting thing about the survival of the combs is that it ranges from 77% to 90%, which we are very happy because it survives. Then the flowering percent of the combs was from 17 to 42.18%. Although it seems to be low, but these flowering of combs comes from those flowerings of the saffron comes from those combs which are more than 8 grams. So it seems to be low, but it is showing very positive result. So these are some of the pictures that I can show you that we are doing the saffron cultivation in Machuca, which is in the border of China. it has a very close glacier proximity to this area, but there is some, we have chosen a slope on the slope of the hill. So the water retention is not much. So this is the best place that we have found so far in our endeavor of expanding saffron cultivation. So some of the achievements that we have been having has been highlighted in various platforms, and the Northeastern Council has also recognized us, and they wanted to collaborate us. So they have already extended support to us, and they have entrusted us to go all out for expansion to all the 7 states of Northeast. So, but having said that, there are so many challenges that we have faced in terms of lack of awareness and expertise, the impact of heavy rainfall in the area. I've been telling in Northeast we are having more than 200 or 2,000 millimeters of rainfall. Then the lack of research and testing facilities. So I would like to invite the research organization to collaborate with us in terms of propagation of the combs. Especially, we know now that there are many places which are suitable, but the lack of the combs is one of the major hurdles that prevent us to expand further. So these are some of the places, like in Meghalaya, we are collaborating with the KVK. So this is in Thangsning, which is near Shillong. This is in Arunachal Pradesh, Shergaon and Mechuka. This is in Sikkim, Sajong and Yoksum area. This is the field survey. We have been continuing doing the expansion process, and recently we have surveyed other places in Northeast, like in Sikkim and Meghalaya and Nagaland and Manipur. So these are some of the photos that we have visited. So in whatever we can, we also try to give trainings to the farmers in the area while we go for survey, along with demonstration. So our target for the next couple of years is we wanted to cover 20 acres of land, covering 500 farmers. So this is our immediate plan as of now. We have started all the groundwork, all the collaboration and all with the state government and other stakeholders. And thank you so much. That's all I can say.
Thank you, Dr. Collins. Congratulations. You said you are not doing research, but this is no less than research. Actually, we researchers, we need you guys to take all the findings to the field, whatever I said in the beginning, from Thali to drug development, that cannot happen without you. Right. So I'm sure we have had such a pleasant trip today from Kashmir to Northeast, right? Holding saffron, holding saffron in our minds and in our, not in hands physically, but in our thoughts and in our mind. I think everybody has enjoyed, but I would like to invite now Dr. Sharma, Director of NECTAR, who is instrumental in bringing all this research knowledge to the field and to the public. Dr. Sharma, are you there?
Yeah, Dr. Renu, I'm here.
Okay.
I'm in a field in a very remote place. So
Are you driving? I saw you with the belt. Yeah, are you driving?
No, I just came down. I came out of the car. So now I'm in a plant, basically.
All right. So we welcome you. And then we look forward to your comments and your experience and your initiatives on saffron in Northeast. Dr. Sharma, please.
Yeah, thank you, Dr. Renu. And thank you all distinguished. I was basically, I felt so fascinated at listening all the talks, because since I was in CAR, so I could have the opportunity to listen all the talks. Particularly, Professor Dhar mentioned about more details about the research. I have a lot of questions. So these questions can be, maybe I can directly talk to them and then visit their labs. Basically, when Dr. Nasreen and Fayaz was talking about that, how it is basically important for the medicines, and it's very important for all of us to know about that, because we are basically a layman, and I'm particularly layman in this particular sector. But why we initiated this particular project, let me just share with all of you, because all the people who are expert in saffrons are here, that we have the technology at this moment. We know that this geospatial technology is there. So we initiated the whole concept. Actually, Dr. Colin told about more details about that, but actually we started whole concept in 2020, just COVID time, because we know that we are just sitting idle, we're not doing anything. So we thought that why can't we take this possibility to expand this saffron cultivation in Northeast. So we tried to understand the whole the environment and soil condition of this through satellite data, and then we just explore all throughout the Northeast, and we found not only these places, 17 different places are like almost similar soil and environment condition compared to Kashmir. 17 different places. This includes Sikkim and Arunachal and Meghalaya and few other places. But as Dr. Colleen was mentioning, that only we initiated in few places, and we found very good successful the way that we thought that better than that. Now I would like to invite, because this is the possibilities, as somebody was talking about the global market, and we are also importing a huge amount as a country. So why can't we take it up in a more details? So I think with this the initiation that we have done in Northeast, and the possibility of doing it there by doing the pilot one. So I found that it is very important for us to take it up further, and I also thank to the Government of India, particularly Ministry of Donor, to initiate some support to us to take it up in a bigger way from this year. And thank to AIST team to initiate this particular collaborative research work, so they can also. And I also welcome all the other institute who are present here in this particular meeting to come forward and do the quantitative and qualitative testing of the Northeast saffron, so that the purpose of doing research could also come out of this whole the concept. That's the main idea. And I think with the kind of the soil, with kind of the conditions that we have, we can do it. But another important point I would like to mention here, that which is also interdisciplinary in nature, we have initiated some approach to that. As Dr. Kalin was mentioning, because of this high moisture content, the combs get rotten with time. So we try to initiate some IoT-based sensors to understand the life of the comb in a regular basis, daily basis in the desktop. So that's another the technology-driven concept that we have initiated, and maybe the expert who are present over here, they can come forward with some more idea. We are not doing in-house condition. We wanted to make it in outside itself. So we are most we are open to collaborate with all the institutions who are present over here to understand more about the Northeast saffron. And thank you very much, Dr. Renu and Dr. Shunil, for giving us an opportunity and giving us opportunity to collaborate with the AIST particularly. And we are looking forward to take it forward in a much bigger way. Thank you.
Thank you so much. And it is our pleasure to serve as a bridge between Kashmir and Northeast, And I think one of the first things we need to do just after this CAFE 100 is to make a NECTAR research consortium. And I would like Dr. Dhar and other experts, invited speakers, to just comment, and Dr. Pati as well, to comment just like, 1 minute each on this collaboration.
Dr. Reddy, just can I just ask one question to Dr. Dhar? Just one.
Yes, please. Dr. Dhar, there you are with the questions.
So I heard that there was a National Saffron Mission, which initiated a long time back. I would like to know the status of this National Saffron Mission and how it could be helpful for us to take it up.
Please unmute yourself.
Yeah, actually, this saffron mission was there, and it was mainly focused at SQUAST, that is Sher Kashmir University of Agriculture Sciences and Technology. And that time we were doing parallelly some work, but we were not associated with the saffron mission at that point. But later, I think almost about 2 years after this mission was in operation, DBT started a network program, and we were a part of that DBT network program. So I think that Saffron mission part, which was with SQUAST, I am not sure whether it is still continuing, but since last maybe 4-5 years, we have no information about that. But our network also was a kind of, it was not so much funded, but I would say definitely it was a handsome money which was given for our network as well. But it also continued, and it culminated somewhere in the year 2022. And we have now in the process of getting another grant from DBT. So, but regarding Saffron mission, I think it's all over, maybe many years back. That's what I think.
Yes, sir.
But
Yes, sir. It's
I may not.
over. Safdar, yes,
Okay.
it's over. I think
Yeah.
I think Saffron mission is already over.
Yeah. Several years back, I think I have no
Yes.
information.
Yes.
Okay. We got the answer. Now, before I address a question on the chat, which is from the Dan Bhatt from Anisha Group, he is our guest, attendant today. And what he's writing is, we look forward to financially collaborate with the Saffron ecosystem. So this is a good news. And what I'm going to do is to make a Saffron consortium with you experts and people who are motivated to do saffron research within AIST and in Indian side, and let's see what we can do and what can be done in India. And you probably didn't think about it. We here in AIST is the far northeast of India. If Shillong is the northeast, we are a little bit further northeast as well. So anyway, I will be in touch with you all and follow up on the comment. This is very, very positive. Thank you, Dan. You want to say something very quickly? You are very welcome to say before I ask the other experts to say comments on the research. Dan, are you there? Dan is a friend and he's from Kashmir, as you can see from his name, Dan Bhatt. Dan, are you there? You want to say something? He probably cannot speak right now. He's not in a environment to speak. But anyway, so Dr. Pati, we're waiting for your comments.
Am I audible right now?
Oh, you wait. Yes, Dan, we can hear you. You want to say something in less than 1 minute?
Yes, namaskar everyone. Thank you very much. My name is Dan Bhatt. I look forward to financially collaborating with the Saffron ecosystem. I thank Renu and the rest of the team, and of course Dr. Sunil Kaul for setting up this wonderful event. And I look forward. Thank you very much, everyone.
Thanks, Dan. Thanks for coming today, and thank you for the comment. I'll follow up on that with you. Dr. Pati, you want to say something within 1 minute? And other people, please switch on your cameras if you want to be in the picture frame. Yes, Dr. Pati.
It was a pleasure and a treat to listen to others. I mean, excellent choice of crop. I mean, saffron interest to India, and I'm happy that Japan is also taking equal interest. And I think both partners can come together and do some fantastic science for this particular review. I'm very happy that many of these science were discussed today, from the biology of saffron to its cultivation, to smart saffron, to medical applications, and extending it to new areas. It's an excellent effort, and I really congratulate Dr. Renu, Dr. Sunil for bringing all of them together. Thank you.
Thank you, Pratap. Thank you so much. So everybody, please switch on your cameras so that we can capture you for today's presence evidence. And this will be put on the thanks card circulated to all of you. Ajay, you want to say something quickly as people are coming up on the video?
It was a very nice event, and I congratulate all of you because this is the 100th cafe series, and a very important topic. And not only that, this generate lot of questions and lot of opportunities for many people. So it's a very
thanks. Thank you, everyone. Thank you for coming, and please keep coming in the future. And I'm going to have now a few screenshots from your, for the group picture. Here we go. 1, 2, and 3. Smile, everyone. This is the frame 1. We had actually over 100 people joining today, There were 88 entries, and there were groups there. Hanyang University, as usual, is in a group of about 20, and GNDU is more than 20 people. I see them all together. So thank you, everyone, for keeping on coming and motivating us to keep this program on. And I think today was very enjoyable, and we had very good comments from everybody. And thank you, Dan, for coming, and we will follow up on these. Thank you, everyone. Thank you. Thank you very much.
Thank you, Manan.
Thank you.
Thank you.
Thank you so much.